In our first experiment, we will be taking flies and exposing them to alcohol on the ground and monitoring the recovery of them both in flight, in altered gravity, and on the ground.
By doing this, we're able to find out whether the alcohol dehydrogenase enzyme is altered in microgravity. After that, we're going to test the recovery of fruit flies and do a biochemical assay to really see how quickly those fruit flies metabolize the ethanol.
For our second experiment, we will be exposing flies in flight on the plane during the altered gravity to determine whether or not altered gravity has an effect on the uptake of ethanol.
If we get some different results, if the fruit flies do end up metabolizing the ethanol faster or slower, it could answer questions about enzyme activity in microgravity.
This is applicable to the ISS because the astronauts may need to take pharmaceuticals or other drugs while in altered gravity.
That could be related to enzyme activity, and so we'll really be able to answer some major questions.
For our experiment, we're using Chlorella algae as a model system to maximize photosynthesis in microgravity. We have designed cubes out of Lexon to grow our algae in.
We're using Chlorella because it has the highest photosynthetic pigment content of any organism known to man right now. It has so much chlorophyll that in fact it's even beneficial to consume it. It'll provide radiation protection to the astronauts on the ISS if it gets there. So it could be used as a potential food source as well.
What we're looking at it using a solid growing environment called agar, which is the gel in petri dishes, instead of liquid media because of the strange behavior of liquids in space.
If our experiment got there, it could be grown both to remediate CO2 through photosynthesis and produce oxygen. It's a free ecological service that Chlorella could perform on the ISS.